to this question, can the Bible improve our science? There's a formation right here called, I don't know if you can see that green thing, the Navajo sandstone. It's a band right near the top of the Grand Canyon. You can see this white band right here. And here we're seeing a close-up. It's about 300 feet thick of sandstone. And the only fossils in there are fossil animal tracks. So what were these animals doing walking around on the sand? Well, a crucial issue here is, how was that sand deposited? And to most geologists, the answer they would give, see, why is this not?
It's got a window.
Can you shut that off? Okay, in a desert, and it's believed to be a desert deposit. The sandstone was deposited, sand was deposited in a desert. And in the desert, the wind blows this way, and it blows sand and makes one layer after another on the side of these dunes. And so here's sand from underneath that has been deposited the same way. And we call it cross-bedded sandstone. And it looks like this in the actual, when it's sandstone. And the tracks go up these slopes, and so they would be going up a slope like that. And then they're getting preserved. Is that going to happen? Well, I read some papers, and they all agreed it was in a desert, and I wondered about that, because it's in a part of the geologic column I would think would be in the flood. Now, maybe the flood was very complex. Maybe there was some wind sometimes. I don't know. But anyway, I wanted to investigate what are the possibilities of how this could have been formed. And there are possibilities. One, of course, is that it was a desert. But another, you see these sand dunes? See, that's not a desert. That's under the ocean. Water and wind does very similar things to sand. So these are underwater sand dunes. Actually, they're usually called sand waves. And so animals can walk, the way they can walk is quite different underwater. And so there are possibilities we need to consider. So is it likely that these animals will walk on these desert dunes and it will be preserved? That's a big question. And I had my doubts. So let's ask you this question again. How can the Bible influence scientific research? And most people will say, well, it can't. It'll just bias you. Most scientists accept naturalism, no miracles ever. But how will this affect their science? Could that bias their science if you make that assumption? If we believe the Bible, how will this affect our science? And anybody can be biased. Anybody can use their philosophy to bias their science. It isn't so much that one can bias and one won't. Bias is a human problem that we all have to deal with. And people like Chadwick and myself and Susie Phillips, we don't just look at the Bible and that's all. The advantage, one big advantage we have is that we understand both philosophies. We understand how the other scientists, what kind of data they use and how they interpret it. We understand how we would let the Bible give us some clues that there hasn't been millions of years and there has been a geological catastrophe, the flood. So we understand both of those points of view, and we're always looking, where can we test between those two? And so I would suggest to you that the Bible can be actually an asset to us in science. And so can we give an example to show that this actually works? So when we're studying the coconut sandstone, there are certain questions we ask. And this is an important first step for science. The questions you ask. What questions are you going to think of? And what questions are you going to try to answer? And so our questions are, how are the tracks preserved in the desert which formed over millions of years? That's one question. How would you do that? Or even, was it a desert? Or could it have been during the global flood? And this is a question that almost nobody else is going to ask. We are asking that question. We open our eyes to think of two things. So here I am, back when my hair was a different color, looking at the fossil tracks here in the Grand Canyon on this sandstone. Collect a lot of data on the tracks. And here's what we find. These tracks are really quite beautiful little tracks, preserved in the sandstone. You see you have the palm prints, and then you have all the little toes preserved. Here are some larger ones, and they're not as clear, but even here you see the individual toes. There's a really tiny one here, you probably can't see it. And so you have a lot of nice details preserved in these tracks. So how do you do that in sand? Well, we did some experiments. A graduate student made this sand dune for me, and then this is in an aquarium under the water. So we could put these animals in either condition and let them make tracks and see what the tracks are like. And here's what we find. These are the fossil tracks, so compare these now with, these are on a dry desert dune. You notice the sand, it's on a sloping surface, the sand slides back and fills in all the details. And so it really is not anything like this. This is underwater. This is the only condition that gives you tracks that are close to the fossil ones. You have a lot of nice details preserved. Now, the geologist who originally defined the Coconino as being desert, he did experiments like this. And, but he didn't do any underwater experiments because somebody told him animals underwater don't walk on the bottom. So he didn't do these. He did these and some others. He decided that the desert, the dry sand, was the right condition. How he came to that conclusion, I don't really understand. But that was his conclusion. But this is what I found. This is best. Underwater is the only one that gives you good tracks. And so we published a paper on that, which actually was picked up and republished in another journal called Benchmark Papers in Geology. That was kind of exciting. But there's more than that. There's more. There are a lot of tracks that, like what you see on these slabs. This is about 12 feet high. And here's a detail of it. And you see trackways going this way. And incidentally, it's on these sloping surfaces, and the tracks are almost always going up the dune. Why is that? Well, that's an interesting question. I don't know the answer. I have an idea. But anyway, so this is a normal kind of track. The animal is just walking up the dune. Here's another one like it. But look at these. They go across, like this. Okay, and these ones that are going sideways, going crossways, they have some very unique features. The animal is, in all cases, all these tracks, the toe marks are pointing up the dune. But these are going sideways. So how does an animal have his toes going this way and he's walking sideways? Have you ever seen animals do that? They don't do that. So how do these get formed? Well, I tried to figure out how you could get an animal going sideways on a dune. Here's no more detail. See how the animal, the toes are all pointing up this way, but he's going this way. And here's another one. This is rather extreme. They're almost going at right angles to the direction where the toes are pointing. So, and there's a lot of these. So how does that happen? Well, it occurred to me, if the animals are underwater, this could happen because whether it's wind or water, the current doesn't go along the surface of the sand. It kind of goes over the top of the dune and then up like this, and so it isn't actually affecting the surface. But you can have gentle lateral currents, sideways currents, moving, which could move the animal sideways. Okay, so what if that's happening? The animal's trying to go this way, but the current is moving him sideways. Well, we were able to study that in this device here with current flowing here and a camera recording it. And in fact, it does happen. The animals sometimes get drifted sideways, and they're trying to walk forward. When that happens, this is what you get. Here are drawings of the actual tracks that were made in the sand, and this is where the animal is. And when they're drifting sideways, they make tracks just like those fossil tracks. Toes pointing this way, but the animal is moving this way. Here's an illustration from the paper that I published on this. And each of these trackways are traced from an actual fossil trackway. And the drawings of the animal, that's my interpretation of what's happening. So here you have animals walking up this way, then he goes sideways, he's still trying to go this way, but the current is pushing him sideways, and then the current stops, and he goes up. That's my interpretation of what's happening. The current is drifting these animals sideways, and that explains all of these trackways. So that's another piece of my evidence that if it's underwater, this can explain these sideways trackways. But there's more. It gets more interesting as you dig into this. Okay, what if I saw something like this? What if this happened to you out in the desert? You've got to start somewhere, okay? So where did he come from? And what if you found fossil tracks like that? And in fact, we do find fossil tracks like that. Now, if you got wings, it's easy to explain this. You land and walk. But what if you don't have wings? And these animals that make these tracks were not winged creatures. They were four-footed creatures. And we do find fossil tracks like this. Here's one. It's a beautiful, smooth slab of stone, and very nice trackways. It starts here and goes this way. You should have had tracks up here, but there aren't any. Here's another one. Trackways going this way. There should have been tracks back here, but there are none. Where did that animal come from? Well, it depends on your environment. If it's in a desert, I have no idea how you could explain that. There doesn't seem to be any explanation. And those who criticize my conclusions have never even tried to explain it. But if it's underwater, that's easy. You swim down and walk, and then swim up again. So again, underwater is the only condition that will really explain this. And here's the nicest one I have. The animal's walking this way, then he disappears. And then either the same one or the same kind appears up here and continues walking. So you definitely have these tracks where you need wings, but they don't have wings. And so underwater is the only way, it seems, to explain that. And then we've been doing other research as well. This is a slab of sandstone that we found out in Arizona and brought back in our sequoia, back to Loma Linda. It's on my office wall. You see all these ripples, current ripples? That's on this sandstone. Well, wind in the desert doesn't make ripples like that. Those are underwater ripples. So that points again to underwater formation of this sandstone. There's a whole lot of other things we're studying. There are dimples in the rock, usually kind of scattered everywhere, and they've been explained as rain prints. They've been interpreted as raindrop prints, saying this head can't be underwater, it has to be in a desert. Well, we find a lot of cases where the rain prints are in rows. And where I come from, rain doesn't fall in rows. There's something else going on. These are not rain prints, there's something else, and we have a growing evidence to point to that. There's something else going on, like maybe little bits of fluid, of water, coming from underneath the sand up through the surface and making these prints. So more and more evidence we're finding that says this is not desert, this is underwater. Okay, let's come back to our original question. If we believe the Bible story of creation and the global flood, here's what happens. We think of new questions to ask, which we would not have thought of otherwise, and which other people are not thinking of. They're not asking, is this a desert or is it underwater? Because they're so sure they know which it is. And it opens our eyes to think of new ways, different from those who believe in naturalism and are not asking these questions. Then the questions must be answered with quality science. So the Bible doesn't say anything about the Coconino sandstone, but it tells us about the flood and earth history, how it's been a short time and has not been evolution and there has been a flood. And this background allows us to think of these new questions, which other people are not asking or not thinking of. Then we have to do careful work to answer those questions. Okay, so we use this unusual approach for science. Does it work? Can we actually get papers published when we do this? Well, yes, we can. even if we're asking questions that may seem unusual, if we're doing careful work, we can get papers published in some very good journals as well. And not everybody out there likes our conclusions. Definitely some of them do not. But if we're doing careful work, we can, sometimes with a little bit of struggle, get papers published in scientific literature. And this is a quality control factor. It's important. Other reviewers have to other scientists have to read our manuscripts and evaluate them. Did we answer the question? Do we have the data to support our conclusions? And yes, they decided we do, and so we got published. So some conclusions. A biblical worldview suggests new questions to ask. And those can lead to new research that nobody else is doing, that others have not thought of. And the evidence indicates the Coconino sandstone formed underwater. Okay, these are here we're dealing with religion. We're helping let the Bible help us to think of new ways to looking at it. But this is just strictly science. The data we collect is science. And then we say, well, perhaps this was during the flood. Now, our science can't demonstrate the flood. This is a religious insight which helps us to come up with ideas of how this all worked. And some of us have been doing this kind of research for years, and this is not just an isolated. I'll give you one example. I could spend the rest of the afternoon through the night, but I think you'd get tired. So I'll use this one example. But a couple other points I want to make. One is, this kind of research, we find, helps to encourage people to recognize that we're not being off the wall. We're not having these crazy ideas from the Bible. When we put it to work in science, it does work, and it helps us to get better insights into science, to understand the scientific data. So that's one advantage. There's another advantage of doing this kind of work. Through the years, we run into and work with various kinds of people, including atheist geologists. And they wonder about what we're doing, and we are able to get to know them as persons. And I could give you, again, many personal experiences, interactions with people like this, and I'll mention one. This is called the Navajo sandstone. It's a similar sandstone to the Coconino. It's also believed to have formed in a desert. And this fellow, his name's Dave. He's a geologist, an atheist geologist. And he had done some work in this Coconino sandstone. And way back when I was just starting, he called me up one day and talked about what I was doing. He had questions to ask. And we argued in the scientific published literature a little bit about these tracks. But one time I ran into him, and I had opportunity to answer his questions in the journal that got published. And so one time I ran into him at the meetings, the national meetings of the Geological Society of America, and we chatted a bit. And he said, I still don't know how to explain your trackways. And I appreciated his kind of open attitude, and I said, well, I don't know how to explain some of your evidence either. So. And his open attitude encouraged me. He didn't just make fun of me. And so I asked him to come to Loma Linda and give us a seminar on some of his research, which he did. He's doing some good research on this sandstone, even though I don't agree with his conclusion. And we took him to a museum in LA. On the way back, he started asking questions. He said, I know there's something different about the way you people deal with faith and science, and I'd like to understand it. And so we got into a very frank, honest discussion of the kind of things I've been telling you about, how we let the Bible direct us to new questions. And he listened carefully, and when he left to go home, we left as good friends. And the next year at geology meetings again, I ran into him, and he started the conversation. He says, your philosophy of science approach still intrigues me. And so, again, I got my courage up, and I asked him, would you like to go out to the desert here and spend a few days with me just looking at the evidence together? Well, he did. So he wanted to, and we did. We spent several days in the Navajo sandstone and in the Coconino sandstone. And we not only talked science, but we had come to trust each other as scientists, and this led to openness in other things. And he started asking questions. What is salvation? What is heaven? What is hell? What is, he wanted to know if you have to belong to a certain church to be saved. And I'm sure he asked that question because his wife is a practicing Catholic, and she's no doubt telling him, no, you got to join the Catholic Church if you're going to be saved. Well, he's not sure what being saved is meaningful anyway, but we had very beautiful conversations, spent hours sitting under a juniper tree talking about these, with him asking questions and me offering what I think are good answers. The third day, we pretty much finished what we were going to do, and I could have driven home that evening, but he wanted to stay another night, camp for another night. And he offered to pay for the campground. And so I didn't know why he was so eager. But anyway, that evening, again, it was hours of fascinating conversation with him. Talking with this open-minded, searching atheist. And finally, it was time to leave, and I was headed for my sleeping bag. And as I got in my sleeping bag, my prayer was, please, Lord, no more. I can't take any more blessings in one day. So those kind of opportunities are precious, and there are many of them that I could tell you about. And what I'm telling you today doesn't just apply to the Coconino Sandstone. We've done various projects through the years with the same approach. The Bible opens our eyes to see things that other people are not seeing, and we end up with publishable research. One big project for about 10 years with fossil whales in Peru, and it turns out they were all rapidly, catastrophically killed and buried. Another project for another decade, thousands of turtles in this formation here. Again, killed catastrophically in different episodes. Art Chadwick and I have been doing a project in Utah looking at rocks and evaluating the amount of fossil burrows in the rock. Now, why would we care about that? Well, the way sediments form today, animals burrow through the sediment, and it should be just full of these burrows if there was enough time. But when you look at these rocks and measure up, and examine them carefully from top to bottom, and we didn't actually go straight up this cliff, I'll assure you. But we find there really isn't much of this burrowing in the rock, which tells us there wasn't very much time. That's another project. Another project we're working on right now is this. This formation is kind of soft mud at one time, which formed into rock. And then this came on top, and most geologists think after this one formed, there was 10 to 30 million years that passed before this one got deposited. Do we find evidence for that time in there? Well, no, clearly we don't. We have paper now ready to be submitted. And so there are many of these projects, and in every case where it's been done carefully with letting the Bible open our eyes, it's been successful, and we've found evidence that indicates the Bible has a much better understanding of this than other geologists do who are not willing to look at that. So God has led us to these projects and helped to open our thinking with the help of some of our colleagues that came before us. And it's very productive and fascinating work, and we thank God for the opportunity this gives us. So, thank you. Okay. In the other side. No, that's all right. That's ours. I don't know where.
Wrong spot. Other cord is blocking me. I should go over here.
Can you hear me? Maybe if I have my head up. My name is Susie Phillips, and I am the chair of the Earth and Biological Sciences Department at Loma Linda University. I've been here before, and I very much appreciate the opportunity to speak to you. So I'm going to talk with you just a little bit about challenges the naturalistic worldview. As we discussed this morning in church, that essentially there are a couple different ways to look at the world. That there is only natural laws at play, or that there has been someone supernatural also in human history and in this Earth's history. As Seventh-day Adventists, of course, we believe the perspective that there is some supernatural intervention in our origin, supernatural intervention in our judgment, supernatural intervention in the world history that we can see preserved in the rocks and in nature. So it disturbs me a great deal to find that there are Christians who will begin to learn a little bit about science, and as they begin to learn just sometimes a little bit, like what they read on Yahoo News, they decide that Yahoo News has told them that the Bible is incorrect. They found this one particular point in science that contradicts what the Bible says, therefore they give up all the Bible. And maybe you know people like this, maybe you have friends like this, maybe you have family like this. However, I want to point out to you and to those folks who are in that position that there are many, many things in the natural world that contradict the evolutionary model. Although most evolutionists don't give up their ideas, they don't give up their belief in the evolutionary model, they just say there's more research to be done. And I would like to say, as a scientist who also believes the Bible as it is written, that there was a creation and it happened in 6 days, and there was a Sabbath and it was the seventh day. And sometime after that, there was a fall, and there was people being brought into sin, and God passed judgment, and there was a flood. And all of this happened in the relative recent past, in a few thousand years ago, not millions and millions. And I will say that this is absolutely what I believe, and there is in fact evidence for this in the natural world. And there's evidence that contradicts the evolutionary model. So I want to just highlight some of these. I'm going to go through them just point by point, but hopefully not too quickly. But to me, the most important evidence is cellular complexity. I'm a molecular biologist. I study bacteria for a living. And I love studying bacteria because they're so much simpler than everything else in the biological world. And this is, in fact, what I study. This is a blown-up picture. If you see that oval there representing the bacteria. Bacteria, some of them are able to swim around in their environment. And so I study their ability to swim around. Well, I don't study the motor. The motor is what actually does the locomotion. But tell me how good a car would be if you had a motor but you didn't have a steering wheel. Or if you didn't have a brake or a gas pedal. So you have a motor and it's just going, but you can't tell it where to go or when to go. So what I study is these things over here that are giving the information to the motor about when to go and how to go. So the motor is important, the motor is amazing, but unless you have a steering mechanism, then it's also useless. The motor is not functional to produce anything that would be advantageous to the bacteria. The bacteria don't have any advantage if they can't steer. So actually I study these steering proteins. Here's a little blown up picture of this motor. The motor, as you may know, is a point that a number of creationists who are not Seventh-day Adventists make reference to. That this motor is so incredibly complex, and the pieces that you find here almost identically mirror the pieces that you find in an electric motor. And that if you find all these pieces and they're all functioning so well, how could that possibly have evolved? Because if you miss one of these pieces, the whole thing doesn't work. And so a creationist, Michael Behe, who's not a Seventh-day Adventist, but talks about this as being impossible to have been produced by evolution, because you can't have just pieces of it being made without the whole thing being functional simultaneously. Well, in my world, this thing is also useless without this stuff. So now not only does evolution have to generate all of this and the mechanism to move forward in the world, but it also has to generate the controlling mechanisms. So the car has to, in order for a car to be functional, the only thing you can really take out of your car is your stereo and your air conditioner, right? All the rest of it you kind of need in there. So that's the same way with these bacteria. If they're going to be able to move around, they need all of these functional mechanisms. This is kind of a blown up picture of what I study. I'm going to throw in some pictures here because I find it so exciting. What I study are proteins that sense oxygen and tell the cell to go toward oxygen, because it turns out lots of bacteria like to consume oxygen the same as we do. And so when they find oxygen, they're like, woo, let's go find the oxygen, and away they go. And so this is their mechanism of sensing it. And they actually have in these proteins that sense oxygen a heme, just like we have in our hemoglobin that's traveling around in our blood, that's carrying oxygen around. These bacteria have that too, and they have a heme that sits in there. And when these things become oxygenated, they turn red, just like our blood. And so when I'm isolating these proteins, I've burst open the cells and I'm pulling out these proteins. That protein isolate is a deep red. And so what the proteins I actually get to work with, this little tube here represents like 2 weeks of work. Anyway, it's a lot of work to make this happen. And then I make mutants of it, and you can see this is the regular functioning bacteria, and you can see how red it is compared to some of my mutants, where they still have the protein, but they can't bind the oxygen anymore. So they're different colors. I actually quantify this and look at that. So I've just had to share a little bit with you of my own research studying these proteins. This is kind of a confusing mess. And I'm not going to ask you I'm not going to ask you too many questions about it, but I want to just use this as an example. I'm going to zoom in on this little portion right here, and here you can see the zoom in. Everything in blue is a giant protein, and those giant proteins are basically like machines in a factory. They're carrying out some sort of function. And basically the stuff in red are little molecules, stuff in black are bigger molecules, and then these blue things, 3-hydroxyacetyl-CoA dehydrogenase. That's that one. What are these? These are giant enzymes. Now if you go back to this big mess, you'll see a lot of blue. And these are the proteins that are responsible for keeping a cell alive. Okay, so we call these housekeeping genes. These are just the ones for making the cell stay alive. They're not really the ones that do all the special functions like make a pile of collagen so you can have hair, or make insulin, or sense your blood sugar levels so that you can have... This is not the specialized function. This is just what you need for the cell to stay alive. So if you're looking at this from an evolutionary perspective, if you're looking at this as from a natural perspective, really this 3-hydroxyacetyl-CoA dehydrogenase, this enzyme, it really is not very functional unless it also has this enzyme. And that's not very functional unless it also has this enzyme. And that you can see how we have a cascading issue of these things all had to simultaneously be functioning in order for there to be a cell that was alive. So the basis of evolution is that you have long periods of time and something comes into existence over a long period of time. Well, we can't have long periods of time. This enzyme will not stay being produced by the cell if it's not being used. And so we can't have 1 million years between the generation of this enzyme and the generation of that enzyme. And what we know about cells right now is that they usually they don't keep around stuff that they don't use. They're not hoarders. They don't hoard DNA that they're not functionally using. And so this is confounded by the complexity that you see here in this diagram. Here at the very center is one of the main energy generation pathways called the TCA cycle, or Krebs cycle. Some of you kids, I know my daughter was studying it in 8th grade and 9th grade. She's like, oh, what's the Krebs cycle? Oh, let me tell you. Let's talk about the Krebs cycle. I really love energy generation. I'm just very excited about bioenergetics. Anyway, and this is, see all these enzymes? These are, all these things have to be functional in order for you to get energy from sugar. Well, almost every living thing in the world can get energy from sugar. And so all of these things had to be functional simultaneously in order for you to get energy from sugar. It turns out burning sugar is a lot more complicated than lighting a match. Because lighting a match, sugar has so much energy in one molecule of sugar that is dangerous to the cell unless the cell breaks it apart very slowly. So you have to have a purposeful slow burn in order to get the quantities of energy in little battery packages that your cell can use. This is what's going on inside of you right now. If you had dessert, This is what's happening right now if you ate some sugar. I'm just zooming in on this guy right here. Succinate dehydrogenase, probably one of the most prevalent proteins in the world. It's present in every living organism, and this is what this thing looks like. This is a giant machine that has a heme, just like that can sense oxygen and bind oxygen. It's got iron sulfide, it's got FAD. This is a big, complicated machine. And of course, this is all built up of amino acids that you see here. So this complexity, to me, in my mind, and in my studies, really speaks volumes about how the model of evolution doesn't actually fit the evidence that we find in the natural world. The natural world declares that in order for a cell to be functional, it has to have all of these processes, and there's no evolutionary mechanism that could have generated these processes. The next thing I'm going to talk about briefly is the human nervous system, neurons in the brain. These are human neurons. This is what's inside your brain, the very, very specialized cells. Neurons are probably the most complicated cell inside your body. Some of them can be up to 1 meter long. Can you imagine a single cell 1 meter long? Depending on how long your legs are. Some of us here have longer legs than others. My husband has like 4 feet long legs. He's very long-legged. But those neurons go from the tip of your toe all the way to your backbone back here. And a single cell can have basically a wire-like. You're wired is what you are. And your wires are neurons, and they're neuron projections. These long things are the wires that in some cases will stretch all the way down to your toe. And that wire collects information and sends it to your central nervous system, sends it to your backbone. And there in your backbone, if it's important information, it might get sent up to your brain. If it's not important information, your spinal cord deals with it and says, you need to get your foot away from that fire. And before you even know your foot is in the fire, you're not even consciously aware of it, your spinal cord is taking care of that and said, get your foot away from the fire. And suddenly your foot is back and you're like, what just happened? Oh, my foot hurts. Then you become consciously aware because it takes a little while. So different, depending on the importance of the information, or you might not feel the fact that right now your pants are too tight, but now I just said it, now you realize they are, right? But you didn't feel it before, right? Some things, your brain is kind of filtering what information you become consciously aware of. So very, very interesting and huge amounts of study. It's difficult to study these things because thankfully, modern ethics says you don't just get to pull people apart and see what happens when you pull out their neurons. We don't do that. We do that to crickets. Apologies to crickets, but because we want to know how they function. Most of what we know about neurons comes from doing that pulling and cutting to squid, because they have these giant neurons. Well, not giant, but some really, really thick neurons that you can actually see under a microscope. They're as big as maybe a period on a page, and so you can see them. They're quite big, and you can do some research on them. What's interesting is that these guys that are trilobites, if you drive a mile or so away from here, you will find millions of them for sale. And I'm sure this is where this guy came from, Tucson. This fellow specimen right here was once in Tucson. You can see this is an extinct organism that is found in the geologic column. It's found very low in the rocks. From an evolutionary perspective, that means they're very old. From a creationist perspective, that means that probably they were one of the first things to be buried, basically. So what's interesting about these guys is the complexity that you find in their eyes. Here's a one that has almost 360 degrees. This is a trilobite eye. Almost 360 degrees of visuality around the top of his eye. A really unique thing. This picture is a scanning electron micrograph of a living fly. So this is a fly's eye. So you can see they have very much similarity, right? They're almost identical to each other. And what we know about a fly eye is that each one of these has a lens. They are multifaceted eyes. So we, as humans, have 2 lenses, one in each eye, but flies have a lens in every single facet. And at the bottom of each lens, collecting information is a neuron. Okay? And these neurons, here you can see coming out of here, are bundled together in the optic nerve. And there they go to the fly brain. And so we can do some research. These are all insect neurons, and where you can actually inject some dye and light up the whole pathway of these neurons. So this is the brain of what's inside of a fly. Not the entire brain. This is just a couple of single neurons that are lit up. But you can tell that these flies have complicated cells. They have these neurons. They have neural cells. They have neural tissue. Now, flies aren't intelligent enough to make, atomic bombs or atomic power, things like that, but they're intelligent enough to calculate where to fly and how not to get hit by a fly swatter, which is probably something you and I could not avoid. We're going to get smacked with it when your parent has it in their hand, maybe. So they have to have the ability to do some processing, and all of these really complicated neurons and all of this evolution that was supposedly had to take place must have taken place like as almost immediately at the beginning of the geologic column, if you believe in evolution. So the really complicated evolution took place before we even have any data. There's actually no fossils down here that aren't really that complicated. So that the fact of neurons and these neurons that are likely present in these extinct species that are some of the earliest that were buried by the flood, and from an evolutionary perspective, earliest living, it says that there was cellular complexity at the very beginning of the geologic column. The very, very beginning, when God created, things were complicated, and this is evidenced in the rocks that we see. The rocks that we find say that there was complicated life. There's always been complicated life. Additionally, the very fact that humans are able to do things, our brains do things that evolution and an environment really could not have called for. Our brains do things like calculus and music and carry out high levels of thinking that are not needed just to survive. So the evolutionary perspective and that model is that you develop, you adapt, you have adaptations that are called for or selected for by the environment. And yet I don't know of an environment that would have required us for our brains to be able to do high-level math or linear algebra. Or calculus. We don't need that ability in order to reproduce. And yet we have that ability. And so from the evolutionary perspective, evolutionary psychology and neurobiology is scrambling to come up with reasons why we have this. Why do we have the ability to do this math? And the reality is that they don't really have great answers. They have hypotheses. But our brains can do things that are so much farther beyond what they would needed to have done if we actually showed up by evolution. We do things like love. We do things like play music. And those things are not needed in order to be evolutionarily successful. And finally, the last thing I'm going to talk about is information. Where is the source of this complexity? If you found something that looked like this laying on the ground, you would think, oh, there was a human here, because this thing has structure and function. It's specific for a specific function, and it has structure. And in California, you find things like this, and you can say this once had a function, and you can say there was a human here. There was somebody intelligent acting on this. The same way there was someone intelligent acting on this because it has a function and it has structure. And this is not functional anymore, but we can imagine what its structure was. Have you ever seen anything that looks like this? So, to me, this looks like this. It looks like rebar to me. And rebar we use in all of our buildings, and we use it to reinforce our concrete. And to me, it looks like this. And this stuff right here is what God created to do the exact same thing that rebar does. This is collagen. These are dense collagen fibers. And they look like this when they're all closely packed together, like in your ligaments and tendons, is what you might find collagen fibers looking like this. Here are loose collagen fibers that you find in your skin. So that's what gives your skin strength, is the collagen that's there. So we humans discovered that if you take rebar and build with it, it makes your structure stronger. And because it's being made with steel, steel has a tensile strength that allows it to be pulled and stretched but not break, which is really important for your skin too. Your skin needs to pull and stretch but not break, because it feels nice to have your skin rubbed sometimes. We don't want to produce cracks in our skin just because we're getting a back rub. In our bodies, we mineralize this rebar, we mineralize the collagen. And when it becomes mineralized or impregnated with minerals, it becomes strong, okay? And that's what our bones are made of, mineralized collagen. So we mineralize that with a mineral called apatite. Interestingly enough, we do the exact same thing when we're building. We take this collagen and we mineralize it. And what do we mineralize it with? Cement. And when you have rebar reinforced cement, you have essentially one of the strongest building materials. And that's what most things are built with these days, rebar reinforced cement. And that's exactly what the good Lord built our bones out of. It's just funny that the best science and the best building that humans can come up with is nothing but an imitation of what God already came up with. My question to you is, where did this rebar come from? What is the source of this rebar? Here's a mill that runs metal. And here's a block of molten metal, and it's molten, it's hot, but it's still able to hold its shape there. And then here's a mill that squeezed this metal down, and we have actually getting closer to the size of what rebar is. Is this the source of rebar? This machine? No, not really. I say this guy's the source of rebar. This is my father. My father designs control systems for steel mills. Now, he's not the one who thought up rebar, but he's certainly the one that tells the machine what to do in order to make the rebar. Okay? Where is the collagen machine in here? Well, it's there. It turns out the information, we've got, this stuff will spit out collagen, and if we're talking about cells around your hair follicles, it'll spit out collagen, and out comes hair. And as we get older, we do it better and better, right? Except in some places. So it comes in other places, right? My question to you is, where's the guy who controls the system? Because as we know, this system doesn't show up unless there's an engineer to build the system and to build the software to run the system, right? So where's this guy? Because this doesn't show up unless there's someone to build the system and to design the software to run this system. So from looking at the model of cells, looking at cells, we can see that there is a machinery, there's a factory running there, and it's running fairly well. But the source of information is not there. We know that there is information, the DNA is there, but who made that in the first place? Every scientist will universally tell you, and the entire universal human experience is that you don't get information from something non-intelligent. Information and function and structure come from something intelligent. So there whole books have been written about this, about biological information, how important it is, and the question of there's information out there. Where did that come from? Well, our experience is that information comes from intelligent beings like God. So I want to ask you, is your worldview really outrageous? We really believe that God created. And as Dr. Brand pointed out, it is actually advantageous to his research to have that belief. And it helps him publish more papers to work from that belief. And I want to just give you the example. There were witnesses to these supernatural events. And they wrote down these supernatural events. These witnesses were demanded that they recant, that you've testified, you've seen something, and we don't believe you, and we want you to recant and say that what you saw was not true. And they said, no, it was true. And they were killed for it. Now, I'm a witness, I'm a scientist, that's what I'm supposed to do. I'm supposed to write down what I see and bear witness to you that this is the scientific reality of the world. I've done this research in the laboratory. Turns out those red proteins, they're really red. And if you take away their oxygen, they stop being red. I'm bearing witness to you. If someone told me, now, what you believe about those proteins and them binding oxygen, that's untrue. We want you to recant or we're going to kill you. I'd be like, oh, no problem. Maybe they were binding something else. Die for my proteins. I'm not going to die for my witness. It's tentative enough that I'm not willing to die for it. But these people were. They witnessed something that was so true that when they were asked to recant, they said, we can't. It's true. And we are willing to die for that truth. That is actually very powerful. The writers of the New Testament, they died because they witnessed something that was beyond what was expected. And they died for that. You also are a witness to the supernatural. The Bible says that when we interact with God, we become a new being. And we are born again. And God says, I will give you a new heart. I will give you an undivided heart and put a new spirit in you. I'll remove your heart of flesh, stone, and give to you a heart of flesh. This is testimony. Your own life is testimony to the supernatural that is a reality in the world. So I've just briefly gone through some things that evolution can't account for: the human mind, the complexity, the Cambrian explosion, that complexity being present in the rocks a very long time ago. These things should give us, increase our faith, help us to recognize that our position of recognizing that God is a reality and that God created is not far-fetched. It's not scientifically unsound. There are other things, the origin of life. There are mathematical, chemical, and physical constants, DNA and information molecules, cosmology. Hebrews invites us to have faith. And so from my perspective, I don't think that we're going to find in the scientific world, in the natural world, a microorganism that when you start reading its DNA, it says, God made me. I don't think we're going to find that. Instead, we're asked to exercise faith. God has given us sufficient data for that. Just briefly, this is basically what evolution teaches. That we started as something very small and very simple, and in reality this is the correct size compared to this. So these are little bacteria, what I study, and this is a more complicated cell, and you can tell it really is much bigger. This is more the size of what our cells are. And evolution says that this thing became this thing, and then after a while this thing became more complicated and joined up with lots of other cells and became this thing. This thing has kind of a notochord. It's a little fish-like thing. And that eventually this thing then became more complicated and became this thing. Okay? So this is the idea of what evolution is, and we all recognize it, but you will really never see this in a textbook because honestly, that's really far-fetched. Any person looking at this would say, really? We've never seen anything come together like that. We've never seen anything become more complicated. And so it's usually not presented in textbooks that way. Instead, it's presented as little word stories about this and a little word story about that. It turns out that a universal human experience says that things don't get more complicated. And all scientific experience says that things don't get more complicated. And so if you're trying to pass this process off as science, you have to convince people that it's true. A colleague of ours was going to a meeting at vertebrate paleontology, so it's a subcategory of the geology meetings. And these are people who study dinosaurs mostly. And at that meeting, there was a breakout session for elementary school. And our colleague was interested in elementary school and what they were teaching science. And so she went to that. And basically they said, well, here's the thing. We've got to have appropriate programs that happen in first grade and happen in second grade. So we had to have things that we teach them in first grade and things that we teach them in second grade, and these are for public schools, and things that we teach them in third grade so that they're ready to accept evolution. Those are the words that were used. Because most people, thinking people, are not ready to accept this because this doesn't make sense if you just look at this picture. But if you're told in first grade that it's like this and you're told in second grade that it's like this, then eventually when you get to high school or 8th grade, you're ready to accept evolution. And they were coming up with a whole model of how should we do this? How should science teachers in public schools be presenting science? Because of that, she, being a Seventh-day Adventist, came back and talked to the Adventist church and said, we've got to have a similar model for how to accept creationism. And to some people looking at it, creationism may seem outrageous because you don't believe in supernatural. But we're starting with children who come from families who believe in God's interaction with humanity. And there are real reasons for believing this. And so the Seventh-day Adventist Church has put together a lot of resources, and I just wanted to briefly mention them to you. The Adventist Church believes in the six-day creation, and we are one of the few denominations left that do. Not only do they believe in a six-day creation, but they invest money in studying and understanding a six-day creation and a flood. They invest money in trying to understand and how these processes, in doing research, as well as generating resources. So one of those resources is this book called Faith, Reason, and Earth History. Look at those authors. Yeah. The authors on this book are Leonard Brand and Art Chadwick, who happen to be sitting in the front row. This book is available for free download. It is a textbook. I've just got done teaching all of our graduate students out of this textbook last quarter in the fall. So it's basically a college and graduate school textbook, but it's easily understandable. Even if you're in high school, this is a textbook that you can comprehend. It is the source of study to thousands of Facebook group that one of our students has started with his colleagues in Africa. And there are something like 7,000 people being involved with him in asking questions and having questions answered by their studying of this book. So the church has sponsored this and been involved in sponsoring that. And of course, Leonard and Art are experts in, uniquely, they will not tell you what their degrees are, but both of them have degrees in biology. Leonard Brand got a degree from Cornell in zoology. And Art Chadwick got a degree, was it UCR? Oh no, Miami. Got a PhD in Miami in cell and molecular biology. They both then have gone on to take huge numbers of graduate level courses in geology to the point where they are now geology professors. They lecture in geology. So their background is in both biology and geology. So they are uniquely fitted to write a book that addresses those things. The Adventist Church has also generated textbooks for elementary school. They have textbooks called By Design textbooks, level 1, 2, all the way up to level 8. These you can click on. There's associated labs that are there for teachers who are using the textbooks. Obviously, they would like all elementary schools to be using these. I wanted you to know about them. You can go here. You can search By Design textbooks and at least see what's in them, and maybe do some of the labs. There's a lot of online labs. The online labs are being developed by Loma Linda University, their IT department. So they're really excellent labs that are there. Adventist Church also has something called the Adventist Learning Community, where all of the presentations, we give presentations like this to teachers and pastors. Sometimes we have maybe week-long conferences where we bring in a number of professors and a number of scientists as well as theologians. Those things are recorded and they're all on Adventist Learning Community. It's a little bit difficult to navigate because you have to know exactly, you have to type in exactly the title of the talk or something, but still it's there. One thing I'm really proud of is the work that's done was sponsored by the Faith and Science Council, the General Conference, and is most easily available at Southern Adventist University's website, their origin resources. This is something that I highly recommend. Videos, high-quality videos with actors, as well as PowerPoints, very high-quality PowerPoints that you can download either as narrated PowerPoints or as just a PowerPoint, and you can download the script. So if a teacher wants to be able to teach, they can just use the script themselves, or they can show it like a movie. So here's some... If you look along the edge here, you can go resources by topic, resources for teachers that will be divided up by grade level, question and answers. And here I've just clicked on some of these. I think I clicked on resources for teachers. So here's an actual movie that's in here. Here I've clicked on Earth Science, and here's grade 2 and 4, grade 6, grade 7, grade 8, and multi-grade. And you can click on those and come into, here I've clicked on something else, a geologic column. These are narrated PowerPoints, and you click on one of those. There's also a number of actual movies in here where there's, like I said, actors that discuss what the geologic column is, that discuss what are dinosaurs and how did they get there, that have scripts that have been generated by people who believe, and then they've been vetted by the Faith and Science Council, which is a number of scientists who are asked to serve on a council by the Seventh-day Adventist Church. So you can rest assured that every single word that's here follows the Bible and follows the Adventist perspective on creation and the flood. So I really love this site, and I really love the information that's available. I highly recommend it. And then the last site I'm going to talk to you just mentioned is a Geoscience Research Institute site. That, of course, is for adults, but they have links to a number of movies. If you're at home on Sabbath afternoon or you're sick Sabbath morning, this is a great place to go. And there's not sermons, but movies about science and how it integrates with faith. Just in case you want to look at that. And finally, then, these are some books that I've read that I think are really great. First off, Faith, Reason, and Earth History. Explore Evolution is written not by Seventh-day Adventists, but by a number of creation-believing Christians. And it's a high school level or a beginning scientist level. And it's got a lot of pictures, and it really is graphically very beautiful and easy to comprehend. Contested Bones, this is a really nice one. I found it a page turner. I couldn't put it down. I read it all day Sunday, the whole thing, and a while back when I read it. It's really great. Also written by non-Adventists, but dealing with the human evolution and the evidence there is for human evolution. Anyway, that's all I had to share with you today, but I'm thankful for the opportunity, and I challenge you to take advantage of these resources that have been generated by the church to increase the awareness of the biblical origins, the biblical truth. Thank you. Amen. Can I get my flash drive out of here?
Wow, I get to follow in those wonderful footsteps. Before I start, I want to share with you just a little bit about another book Leonard's written. He's a very prolific author. And I have a copy here in my briefcase, this one. It's called God, Science, and Friends. If you have young people that you're involved with, this is a whole message book. And I'm halfway through reading it to my grandkids. And it goes through every truth beautifully. And is a great book for you to share. And that's by Leonard Brand. There's also one for sharing with friends that believe in evolution. What's the name of that one, Leonard? Creation, ? Really, ? And that's a conversation between a scientist who's a creationist and a colleague scientist who's an evolutionist. And they go on a long trip together. Very interesting. Well, I was not going to talk about dinosaurs today, but I want to say a little bit because somebody asked me to. So I just want to point out a couple of things. This is just a single shot of our museum at Southwestern, our dinosaur museum. We have a lovely museum. I have had thousands of people have gone through and seen it, and nobody yet has come out and say that was a bummer. They love it. People love it, and they say, wow. I heard a girl the other day, it was from Campfire Girls. She said, wow, that's the neatest museum I've ever been in. Well, I don't know if she was from Tennessee or what, but anyway, this is our museum. It's at Southwestern Adventst University. It's free, so you don't even have to pay to go through. It's based on this project in Wyoming, which we started 20 years ago with Earth History Research Center, which is a group of scientists that believe in creation, and Southwestern Adventist University. It's on the Hanson Ranch in Wyoming, which is located right here in Niobrara County. This is northeastern Wyoming. This is Niobrara County right here. And right in here is where we're actually working. This green is the rocks that contain dinosaurs. The brown doesn't have dinosaurs in it. So we've been working there for 20 years. Each summer we take a group of people out there, and some of you may be interested in joining us. If you are, it's a wonderful experience, and students that have been out there, students and colleagues and friends have been out there, can't stop talking about next year. That's what they want to talk about, is next year when I come back, this is what I want to do. And we have over 100 people every year that come out there and join us for a day, a week, or a month. We're there for a whole month of June. This is our field camp. This building was built in the year 2001. It's a 24 by 48 inch foot building. It has served us well for the first 20 years, but it was not built to last 20 years. And right now it's in really poor condition. And we're developing plans to replace it because you can't feed and take care of 130 people in a building that's that small. And so we have developed plans for a larger building, which I'll share with you just because it's fun. Let's see if I can find it. This is our plans for a building in the coming year. It's a much bigger building. It will handle the number of people we have, and I think there's more to it down here. It has a little museum in it, so we can display some of the bones, and tells about the history of the Hanson family. And it allows us to run this project as it deserves to be run. One of the problems was when we started 20 years ago, we thought we'll be out here for 3 or 4 years and be done with it. But the longer we worked, the bigger the bone bed got. And we now realize this is a heritage site. This is going to go on as long as these kinds of activities are carried on the earth. No, we're not anywhere near running out of bones. We know how big the bone bed is. It covers probably 50 acres, and we've excavated about 1 acre altogether. So there is a whole lot more work to be done. So we've decided that we need to leave behind a footprint which is worthy of the project, and so we're working on that right now. Anyway, any of you that are interested in coming out and sharing time with us, we would love to have you. Just a couple more slides here. Wyoming is an exciting place to work. You know, I came from California, and in California, the weather, they don't even look at the weather because it's always the same. It's boring. But it's never boring in Wyoming, I can assure you. Weather is exciting out there. Here's a tornado we saw one day, not too far away. It's also beautiful out there. Spectacular in the month of June when we're there. And very often we see this. Now, you might think, well, that's a once-in-a-lifetime scene. We saw it twice this summer because in the evening when we're in camp, the sun is over here and the rainstorms go to the east. So they're going past the camp and the sun's behind them. So we very often get this whole rainbow right over the camp. I've marked out the spot over here and this spot over here for digging, so we'll see if there's anything to that story. Well, I want to talk about something else, so I'm going to stop there. But I just encourage any of you who are interested, see me afterwards. I'll tell you how to get to our registration site so that you can join us if you wish. Now I want to talk about creation. And I want to pick up some of the things that Susie talked about. But more importantly, I want to talk about a little bit of math because this is after lunch and I know you all are very excited and math is really important. And I want to help you to understand why evolutionists' belief in origins is folly. And I think we can do that very quickly. But first you need to know what they believe. What do evolutionists believe about life's origin? How did the first cell come into existence? Does that make sense? I'm going to talk about that. Life began as a result, this is the evolutionist perspective, life began as a series of accidents, undirected and unplanned. So any idea that you can put a scientist in there and have him do something that's relevant for the origin of life is ridiculous, right on the face of it. Because as soon as you put a scientist in there, you take away the undirected and the unplanned. And it has nothing to do with the way evolutionists think things happen. They say it may have taken a while, maybe even 1 billion years or more of accidents. So next time you want to make something, you start with an accident and then you repeat it many, many times. You're going to get what you want eventually. But it ultimately would have had to have made proteins. We'll talk about what proteins are for those of you. I know some of you this is old hat, but some of you may not know what proteins are. They are the structural and functional elements of all living cells and the nucleic acids that code for them. So we're going to talk about how these things could have come into existence. Well, here's how the model starts. You have air. Okay? Your air and water. And you have meteorites hitting the earth, and you have lightning and energy sources interacting with the elements in the atmosphere. The atmosphere that was chosen, notice, chosen. What does that imply? Planning? Yeah, planning. And so anyway, these are the molecules they put in it. And so these experiments were done very early, 1950s, by Urey and Miller. And what they did was they put those four gases in this closed system. They heated it up, and they drove those gases around here. And then when it got over here, they hit it with a spark. And the spark interacted with those gases, and they were hoping that this would make some small molecules that would be relevant for life. And indeed, after they let this run for a few weeks, they sampled over here, and they found there were some small molecules that looked kind of like this. Now, these are important because this group right here is called an amino group. This group right here is called an acid group. That makes a amino acid. And amino acids make proteins. Well, there are hundreds or thousands of amino acids you could make, but only 20 of them are involved in proteins, all proteins of all animals. So you see a little problem there. If you're going to make amino acids, you better make those 20, all of them. But in fact, they only were able to make just a few of these, just these right in here. And that's all they could make in that simple experiment. Without all 20 of them, you can't do anything relevant for life. In addition, there's another problem. It's called the enantiomer problem. The amino acids, all except for the simplest one, all the rest of them come in two forms. Here's one form, here's the other form. Now you might think, well, those look alike to me, but if you look carefully, there's a mirror plane here. These are mirror images of one another. That means that the molecule you put this into and the molecule you put this into are going to have different shapes. You see that? Because let's say you're joining onto the red thing here. It's going to go off in this direction here. It's going to go off in that direction there. And those are different in the forming of the molecule. So when we make a protein, and here's how you make a protein. You take an amino acid like this one here. You join it to another amino acid like this one. That joins to another amino acid, and you begin to join into these long chains to make a protein. This bond right here is unique. It's a peptide bond. It prevents the rotation of the molecules. So every time you put a peptide bond in there, you can no longer spin it around that joint. That means that when you put an amino acid in the protein, it has to be the right amino acid in the right place in order to maintain the structure of the amino acid. Once those amino acids are joined together, you get a long chain of amino acids. Those fold up, and they form what's called secondary structure, which can be an alpha helix like this or like this. And then that whole thing folds up to make a protein molecule with tertiary structure like this. And you might think that was the end of it, but no, it's not. That tertiary structure has to interact with other proteins in order to make a quaternary structure like this. So here you have a molecule of hemoglobin. That hemoglobin molecule is made of 4 molecules, 2 alpha and 2 beta hemoglobins. And those all have to not only carry out their function, they also have to interact with one another to make that structure. Now, hemoglobin is really amazing. This heme right here picks up oxygen from your lungs, and it carries it throughout your body. When one oxygen goes on, the structure opens up and makes it easier for other oxygens to come on, and so it gets fully oxygenated in your lungs. Then once it leaves the lungs, it goes out into your tissues, it starts dropping off oxygens. But the more oxygen it drops off, the harder it is to get the rest off. So it actually takes oxygen to all different parts of your body without just dumping it in the first place it comes to. It's an amazing molecule. What do evolutionists believe about life's origin? They believe that the first living cell came about through the processes that happened as a result of accident without any planning. To make a typical protein, just one protein inside a cell of about 250 amino acids, that's kind of an average size protein, there are 10 to the 325 different ways you can put those amino acids together. Because each one of those 250 places you can put 20 different amino acids in. So to make a protein with 250 amino acids, there are 10 to the 325 different ways you can do it. If we allow for some redundancy, some of the amino acids can substitute for one another without too much change. There still are about 10 to the 250 power different protein sequences that you can make. So if you want one of these proteins and you're just putting amino acids together randomly, it will happen once in 10 followed by 250 zeros. That's a lot. I don't know, million, billion, trillion, quadrillion, quintillion. I can only go so far. So I can't really give you an idea of how big that is, but we can try. If we take the whole ocean, that's about 10 to the 21 quarts of water, or liters of water, and we put 1 billion bacterial cells per liter, and we allow 20 minutes for those bacteria to replicate, and every time a bacterial cell replicates, it tries a different combination of protein. So that's 1 billion cells in each of 10 to the 21 liters of ocean water. In all of time, that is 1 times 10 to the 14 generations, that's with 13 billion years for the universe, you could have tried 10 to the 50th power times. That's the maximum number of tries that you could get in the whole history of the universe. That looks like this. You see that number? Compare that with that one. And you see how impossible it is, even with absurd conditions. That's only one protein. The numbers, if you consider what is really required for even a simple functional cell, are truly staggering. One figure was 10 to the 8 times 10 to the 10th power. 10 to the 10 is 10 billion. 80 billion zeros. 10 with 80 billion zeros after it. That's to get just the proteins in a cell by random processes. Another source gives 10 to the 50. 10 to the 50 is 10 with 50 zeros over it. Number of zeros, it's ridiculous. How ridiculous is it? If the history of the universe is 10 to the 24 seconds, which is about 1 billion times greater than 13 billion years, and there are 10 to the 90 particles in the universe, and transitions of state, which is the fastest thing that can possibly happen with a proton, if transitions in state take place at a rate of, 10 to the 44 per second, then the entire history of the universe, given all the particles we know in the universe and all the time we know in the universe and the fastest rate of change that we know in the universe, only at 10 to the 150th power events could have occurred. In the history of the universe, you cannot give me a statistic with more than 10 to the 150th power for anything that's happened in the history of this universe. To put it another way, given all the time and all the molecules, we could only have tried out an infinitesimal fraction of all the possible amino acid combinations in protein. Even if we had an endless supply of just the 20 amino acids and a system for joining them together, which we do not have. So let's assume we do have such a system for joining amino acids together rapidly in lengths of, let's just say 100 amino acids. Let's make a protein 100 amino acids long. Can we predict from the amino acid sequence alone what the finished protein is going to look like? Because that's all that matters. The shape of the protein determines what it does. So if I could predict how this series of amino acids would look and how it would behave in the cell, I could be the master of the universe. I could make proteins that would do stuff. So IBM said, let's do that. And in 1999, they started a project called Blue Gene. Blue Gene was, the objective of it was to build the hugest computer in the world. They talked about 1 million processors, all in parallel. And they would run that for a whole year and see if they could solve the protein shape problem. So they built Blue Gene. This is the computer. They put two processors on a card. One processor on a card, 2 of them there. They put 2 processors on a chip. They put 2 chips on 16 chips on the node board, 16 compute cards on the node board, and then 32 node boards in a cabinet, and they had 64 cabinets. That's all one computer. And they built that, and they finally got it done, and they assigned it to solve the problem of how to make a single protein, how to predict the shape of a single protein with 100 amino acids long. They dedicated the computer for a whole year, the whole output of the computer, to solving that problem. Now look, this is a problem if you're a cell, you've got to solve right away. Because you've got to have a protein that does something. You can't try a jillion times to make something. You've got to have it work. So this is a problem the cell, if it evolved, had to solve for every single protein. And IBM built this big machine. Everybody was enthusiastic. We were hopeful that in a year's time, we'd be able to design custom proteins that would do whatever you wanted to do. At the conclusion at the end of the year, they couldn't predict the shape of even a small protein from the amino acid sequence after the entire year of processing. Now, I could have told them that ahead of time. It's just math. But they spent the whole year output on that, and it didn't do them any good, didn't get them any closer to understanding protein folding than when they started. So they decided, well, let's build a bigger computer. So they built Sequoia. Sequoia, by then they had chips that had multiple processors on them. They used 16-core chips. They built a module that had that chip on it. And they put those on a compute card, which had the memory associated with that module. And then they built nodes of 32 compute cards. They built mid-planes with 8 of those. And then they built cabinets, which had 16. And then they built 96 cabinets. Here's what it looked like. Just to give you an idea, this is all one computer. No more discussion about the Blue Gene project, by the way. These were all bought up by the NSA, are used to read your cell phones and stuff like that. But since we've gone this deep into computers, let's go a little deeper. Susie was talking to you about the brain, so let's talk about the brain. The fastest supercomputers were thought to be able to mimic the output, that's Sequoia, was thought to be able to mimic 1% of the output of a human brain. 1%. But that was before they began to realize that in the brain, in the human brain, every neuron is acting as a computer, as a PC. And if that's the case, there are 86 billion neurons. Do your math. It would take 60,000 Sequoias 60,000 sequoias just to mimic the output of one human brain. And you think that happened by accident? What else do you know? Then, just 3 months ago, we discovered this. Susie started talking about this. A single neuron in the cell of a fly's eye, a single neuron, acts like 1,400 individual computers. Here you can see in the brain of here's the eye here. This is the optic ganglion here. Here's the brain down here. This part right here we're going to zoom in and look at. Here is that one nerve coming down here, and it diversifies and spreads over all these surfaces here. Here you can see the ends of it. It's been dyed with a green dye. And there are about 1,400 termini in just one neuron. Now, if we didn't know that half a year ago, and we just found out this, how much more is there about the complexity of the human brain that we don't even know yet? The answer is a lot more. Am I right? This begs the question, how could such a complex system exist without a designer? Let's take a little closer look at how complex systems develop. And for this, I'd like to turn the lights down if you can, because you're going to have to look at up close. We each arose from a single cell. Did you know that? At one time, you were one of those cells. One of those cells that happened by accident. Well, not really. I want you to see what happens when that cell, that single cell that each of you started out as, I want you to see what happened when that developed into an entire organism, you. How can you pack into a single cell all the information necessary to make you? Have you thought about that? Let's watch as we look at this newt egg develop. This is the egg of a newt. It's like a salamander. It develops, it hatches in the water. It forms an organism that lives in the water for a while, and then it grows legs and goes out on land. What we're going to see here is we're going to see cell divisions take place. This is time-lapse, so speed it up a great deal. And you're going to watch the division. Notice the first two divisions are right through the middle, so it gets into 4 segments. The third division divides it into 2 ends. Now, who's telling it where to divide? How does it know it's supposed to make that third division at one pole? And when it makes that third division, it's deciding where your mouth's going to be and where the other end of your body is going to be. That decision is already made after the third division. And then watch as it develops, and I'll make some comments as we go along. Here you can see the egg with its first furrow, first division. Second furrow, second division. Watch the third one. There it is. This is now the head. That's the tail at the other end. Now watch as it divides. The cells get smaller and smaller. All of a sudden, some of these cells decide, we're going to go inside. We've had enough time on the outside, now let's go inside. They're going inside and they're going to produce the tube that forms through your body. Starts at your mouth and ends at your anus. We call it the intestinal tract. That all happens by these cells deciding, I want to get on the inside. Who's telling them to do that? How do they know I'm supposed to be in there? Who's directing this whole thing? Where is the information coming from that makes this happen? And I want you to think about that as we go along. Okay, there's the completion of that part of the life cycle. Now, we're going to develop, down here, we're going to develop the neural shield, right in this area right here. And you watch as the nervous system develops. Okay, there's the neural shield. And now it begins to fold on itself. Forms the nervous system. There it's closing. This is the brain up here. This will be the tail down here. And now you're going to see something that just defies imagination. These cells along here are called the neural crest cells. All of a sudden, those cells decide, I'm going to go be this, or I'm going to go be that. And there you can see them. You see them moving? Those are neural crest cells. They originate up there along the top of the nervous system, and they go throughout the body to various places where they'll form specific kinds of cells in specific locations. Now, if you're an engineer and you were given the job to assign these cells a place to go and tell them how to get there, how would you do it? Where would you start? And yet these all know exactly where they're supposed to go and what they're supposed to do. Let's just watch it go on to completion just because it's so amazing. Here you can see the continued development as the heart begins to beat. You can think about this as you inside the womb, because it's very similar. Notice these pigmented cells on the back. You see those cells? Those each originated as a neural crest cell that migrated from the nerve stem out into the tissues and became those melanocytes. Here's the heart beating. These are the gills here. Those of you who are mothers, you recognize these movements. There's the heart beating. Notice the blood circulating now through all these tissues, where blood vessels decided they were going to grow in order to supply those cells with blood. Here's the gills along here. Those are present only during the water phase. Here's the eye. Notice the circulation of blood in the eye. Now, there's a problem. As it matures, it's captured inside a very tough outer envelope. There's blood circulating through the gills. It's in a very tough envelope. That animal cannot get out of it by himself. In order for him to get out, his head, at exactly the right time, has to secrete enzymes that soften that material so he can get out. You watch it happen. He doesn't break through. He's released. See it disappearing? There it goes. There he goes. This is astonishing to me as I think about the intricacies of development and think about my own self, and that I started out as a single cell. There were instructions in that single cell that were designed to enable it to become a being, a human being. And you've just seen that right here before your eyes. This movie, it's called Becoming, by the way. You can find it on Vimeo. It is an astonishing thing to see, because if you ever wanted evidence that there had to be a designer and that evolution could not have had a role in producing this complexity, can you imagine this happening by accidents over billions of years? You don't need mathematics to convince yourself that life is not happenstance. And to me, this is one of the most compelling arguments for creation that I've ever seen. Thank you.